Right now, it's an honor for me to introduce Dr. Anil Biricik and his talk recommendations for the IVF laboratory for the most effective cell-free DNA analysis during non-invasive PGT. Dr. Anil Biricik received his PhD from Sapienza University of Rome in 2011 with his work on genetics of endometriosis. Dr. Biricik has published more than 30 peer-reviewed articles as author and co-author in molecular biology and reproductive genetics fields. He is a scientific director of the IVF and PGT business unit at Eurofin's Genoma Group, Italia, and his main research areas are the genetics of female and male infertility and novel application methods in pre-implantation genetic testing. Welcome, Dr. Biricik.
Okay, Catherine, thank you very much again for your introduction. And my presentation will be some recommendations for the IVF laboratories for the effective cell-free DNA analysis during non-invasive PGT applications. This is my disclosure and disclaimer. The presence of cell-free DNA molecules in sperm culture media and blastocellular fluid has been demonstrated more than a decade ago, and this opened a new possibilities for the genetic testing of pre-implantation embryo without need to embryo biopsy. And the exact mechanism of cell-free DNA release into sperm culture media and blastocell cavity is not very well known, but apoptosis is most likely cause, and the recent study by Ana Domingo Miles et al. Showed that there are also some mechanical mechanisms during the blastocyst expansion may cause the release of these small DNA molecules in the sperm culture media. And non-invasive PGT applications can bring many advantages. First of all, non-invasiveness and easy accessibility for many IVF laboratories without needing any expertise, and probably potentially reducing the costs rising from the embryo blood biopsy equipments, and especially for the countries where there are some legal and ethical restrictions with embryo biopsy. And also it can be applied on the poor morphology embryos when embryo biopsy is not possible. However, there are also some limitations and restrictions for the potential use of non-invasive techniques, like first of all, contamination risks from various sources and standardization issues. And currently it can be also offered as a prioritization test, and especially it is much more adapted for aneuploidy screening and for PGTA, and it is less effective for PGTM or PGTSR applications. And the detection of chromosomal mosaicism is also challenging. So if we look at the origin and the amount of cell-free DNA in sperm culture medium blastocell fluid, first of all, the one comprehensive study by Rodriguez et al. Showed that the quantity of cell-free DNA in sperm culture medium is expected about to 3.2 until 12.6 picogram. It is more or less in medium if we say the 6 picogram is like a genomic quantity of a single cell, and this is more or less the same amount of cell-free DNA we expect in blastocell fluids. And if we look at the origin of these cell-free DNA molecules, that basically we expect them from trophectoderm or inner cell mass, and but not only, also of course from maternal cumulus cells, polar bodies, sperm cells, or even some exogenous DNA may also contribute the presence of this cell-free DNA. And also we should consider the additional biomarkers like proteomic, metabolomic products or exosomes or microRNAs. Sampling methods in non-invasive PGT basically there are two approaches, and the first is the blastosynthesis. I think next speaker will give more details on this, but briefly, in this technique, the sample collection needs an expertise approach by an ICSI needle entering the blastocell cavity and aspirating the blastocell fluid. And we are talking about like 0.3 or 2 nanoliter of quantity. And this method is a little bit controversial due to its minimal invasiveness. Sperm culture media collection instead is totally non-invasive, and since it doesn't need any embryo manipulation, and it is very simple for collection. But of course, we should consider all the presence of biomarkers together with cell-free DNA in the spent culture media. And another approach is the combination of both blastocell fluid and the spent culture media by zona drilling. In this method, the blastocell fluid can combine with the go out from the blastocell cavity and contribute to the spent culture media. And the concordance studies are showing the highest concordance once spent culture media and blastocell fluid combination applied, analyzed together. But this method also is being considered as minimally invasive. If you look at the workflow of non-invasive techniques, after the ICSI fertilization, embryo culture has been done until day 3 or day 4. And the fourth day of the embryo culture, medium droplet is being changed, and the culture is being prolonged until day 5 or day 6 until blastocyst level. And during this culture, and together with the embryo droplet, one empty droplet, medium droplet, is being can be used at the same conditions as a blank sample. And the blastocyst level embryo, while blastocyst embryo is being vitrified, spent culture media can be collected or blastocell fluid and together with the medium blank, and these samples can be amplified and analyzed by the downstream applications like NGS for aneuploidy screening. In the invasive method, instead, as we know very well, at the blastocyst level, trophectoderm biopsy is being done while the embryo is being vitrified. These blastocyst samples can be analyzed by NGS, and the concordance studies are usually designed for the comparison of genetic results from these both samples, trophectoderm sample or cell-free sample from the same embryo. If you look at the NGS plots coming from the spent culture media, they are quite clear and very similar to the trophectoderm sample, and we can easily determine the euploid XX or XY samples, and also some deviations in NGS plot like trisomies or monosomies, as we can see the partial duplication of chromosome 9, or as we can see the bottom image, the monosomy of chromosome 4, trisomy of chromosome 21, and monosomy of 22. It's quite easily determined. It can be determined easily. And for the validation of non-invasive PGT applications, most of the studies has been done by concordance analysis. And if you look at in the literature, of course, there are only a few of the publications available in the literature, but if we can see, there are many the different applications, different study designs, especially for the amplification methods or culture drop size are different, and some samples also using some studies showing the assisted hatching or frozen solved embryos, and as a consequence, ploidy concordance are changing from one study to another. And if you look at the blastocell fluid studies, usually amplification rate and the concordance rate is less when we compare to the spent culture media. And all these data shows us that especially the standardization and the correct study design for non-invasive PGT applications are essential. So if you look at the challenges for an accurate cell-free DNA analysis, and first of all, DNA contamination is the first issue. And as we know, the main sources for the contamination is the maternal chromosomal cells or polar bodies. If we look at usually basically false negative results coming from the spent culture media are female profile, which is demonstrating the presence of the maternal DNA. Or paternalogeny deriving from the sperm cells may also contribute the DNA contamination. Or it is very rare, but some exogenous sources from IVF laboratory can also cause DNA contamination. And another study is demonstrated by Hammond and colleagues is also manufacturing process also may have some traces of genomic DNA in commercial media. So another challenge was the technical the challenge for the optimization of sample volume and handling. Usually spent culture media volumes are reported are between 5 to 30 microliter, and as we know, these volumes could not always be compatible for the following target PCR or whole genomic amplification protocols, because we expect that if the initial sample volume is higher, then the amplification efficiency will be lower. And if we consider that the recommended initial reaction volume for the PCR or whole genomic amplification is 2 or 3 microliter, these volumes are will have an important impact on the amplification efficiency. And if you look at the blastocell fluid, instead the volume is 0.3 or 2 nanoliters. These are very small quantity, and these volumes are quite compatible for PCR or may have create some problems with the handling. In fact, these the studies with blastocell fluid are showing less performance for amplification. But these may come from the handling of the volumes or some biological factors, as Magli and colleagues showed that if the blastocell fluid amplification is less, then this also showing higher pregnancy rate, or in a good amplification is showing the lower pregnancy rates, which shows the probably apoptotic status of these embryos, and they will have which have less implantation potential. And another challenge is the quantity and integrity of DNA, and the amount of cell-free DNA is, we are talking about 3 picogram, and differently than the other cell-free DNA analysis methods like non-invasive prenatal testing, these small amount of DNA requires an effective whole genomic amplification process. And fragment sizes of cell-free DNA absorbed in 2 populations, like 160 and 220 base pair, and 300 and 400 base pair, as we see in this study. And these also should be considered as a challenging factor for a successful amplification. Or some other methods like some amplification methods like multiple displacement amplification requires a continuous genomic DNA template for the amplification, and this may also limit for amplification of cell-free DNA molecules. If the targeted amplification protocols will be used, then this may require some optimization of the small amplicon designs of PCR and NGS library preparations. So all these factors are showing us the adaptation of PGT and IVF laboratories for non-invasive PGT applications. The first important thing is to preventing the contamination. It is very basic, but some specific precautions should be taken during the IVF lab protocols, like using mask, surgical gown, hair cap, and DNA-free hood surfaces, and fresh adequate of culture media for individual pipettes or for sample aspiration, and so on. Oocyte denudation is very crucial to avoid maternal contamination, as we expect from granulosa cells or cumulus cells. ICSI is always preferred to avoid maternal contamination, and serial media washes during the culture of the culture droplet is very important. And in a recent study, also showed that instead of single droplet rinse, if the sequential rinse, so more than one droplet is being applied, then the concordance rates are being higher. And this is also showing that the the early stage of in vitro development maybe leaves much more apoptotic cells, and it is not very good representative of the embryo. So that's why the culture droplet change and the sequential watches are important. And from PGT lab, of course, we have to consider the contamination detection markers like SNP or STR markers, which can be the indicators for maternal or external contamination, and also blank sample, which is cultured together with the embryo, is important to check the external contamination control. And also data analysis methods like specific NGS bioinformatic pipelines for data analysis and interpretation can be also very helpful to understand eventual contamination. And the volume of spent culture media is an issue for the standard IVF procedure, because we know that in the IVF laboratories and embryologists, they don't want to reduce the culture droplet volume, but it is essential to find an optimized volume for an ideal DNA amplification after cell-free DNA collection. And according to the studies, the 5 or 10 microliter is ideal for effective amplification, but also 15 to 20 microliter is quite acceptable. And if a time-lapse system will be used, and then a culture dish with single wells and separate media drops should be used instead of multiple micro wells to avoid any cross-contamination between the embryos. And if the culture volumes optimized, then both sequential media or continuous single media would be adapted to non-invasive PGT protocols. And from the PGT lab, of course, the volume and the amplification protocols should be adapted to this culture media volume. So in our laboratory, for the initial pilot study, we did some amplification trials with different culture media droplets ranging from 5 to 30 microliter. And after whole genomic amplification, DNA concentrations were higher in 5 microliter and 10 microliter initial culture droplets volumes. And it was less than 20 and 30, but even 20 microliter were giving almost a very good results for amplification. And quantity and integrity of cell-free DNA. This is also very important, because we should know that if we have enough concentration of cell-free DNA, then we expect very good amplification results. And the ideal culture time to collect cell-free DNA from spent culture media is between 24 or 48 hours. And preferably, the better results is coming from day 4 to day 6. For this aim, and our group has studied the concordance analysis study by using 482 spent culture media and the biopsies, the corresponding biopsy trophectoderm biopsy sample. And we observed that the informativity and the ploidy concordance results are better in day 4 to day 6 spent culture media samples. And the DNA concentration after amplification, as we expected, from day 3 to day 6 group was higher. But of course, this is one expected results, because more embryos, much more time remaining in spent culture media, more cell-free DNA is released. So that's why we set up our ideal embryo spent culture media collection time, like period like day 4 to day 6. So this is also another issue for the culture time, to prolong the culture time to the until day 6 or even day 7. But there are some studies are giving very promising results, even if the embryo culture is being prolonged until day 7. The aneuploidy results and the embryo implantation results in day 7 are quite similar to day 5 to day 6. So this should also be considered as prolonging culture the culture time shouldn't have any negative impact on the embryo implantation. So another important factor is span culture media integrity. Because we are talking about very small cell-free molecules, DNA molecules, so we are very sensitive for the degradation. So as soon as media sample has been collected, they should be stored in -20, or they should be amplified immediately for library preparation. The fragmented structure of this cell-free DNA molecules are also oblige the genetics laboratory to adapt their amplification protocols. In fact, some small modifications or also for the amplification protocols has been done and should be validated. And after our validation study for the concordance analysis, we applied our method for 68 span culture media cases coming from the blastocyst level embryo, just to understand if there was a difference between day 4, day 5, or day 4 to day 6 amplification rate and informativity rate. These are very not a big number, are small numbers, but we saw a very similar amplification rate and informativity rate between day 4, day 5, and day 4, day 6 groups. This means that the 24 hour and more, we can get quite satisfying results for amplification and informativity. Another factor we just checked if there was a difference between the medium brand. We tested the 2 different medium brand, but the one is about 55 samples and another was only for 13 samples. So the numbers were not equal, but even using the different span culture media samples, we observed a very compatible amplification and information rate. And another parameter we checked for the performance of non-invasive PGT was the embryo morphology. Of course, we classified the embryo samples like grade A, AB, or BB, or grade C. Again, here we also observed the cell-free DNA analysis performance was quite similar between each groups, and especially for grade C group, there was only 7 embryos, but they gave 100% of amplification and informativity rate. So it is very small number. So statistically, we cannot say, but we expect that, of course, if the morphology is not very good, this may also be an indication of the presence of apoptotic cells. So that's why maybe we get a very high rate of amplification and informativity. So the main question is if may cell-free DNA analysis culture media replace trophectoderm biopsy. Of course, there are some studies, promising studies, is showing that higher sensitivity and specificity rate when, for instance, in this study, span culture media and trophectoderm biopsy samples results as being confirmed compared with the whole embryo results. And the non-invasive PGT approach was giving a higher sensitivity and specificity than trophectoderm biopsy samples. But of course, this was only 50 samples, and it was a frozen thought cycle. So a little bit, this can be criticized, but like this is not a real scenario for the routine IVF practice. And another sample, another study is only with 10 samples. Again, non-invasive PGT sensitivity and specificity were higher in the non-invasive PGT group. And another study from 2019 by Carmen Rubia and his colleagues showed that clinical pregnancy and ongoing pregnancy implantation rate was higher in the sample group where spent culture media and the trophectoderm samples were both haploid, and the less the performant clinical data has been observed if the spent culture media showed an aneuploid and trophectoderm sample was haploid. So this is also very small number, but can be a good indicator for the performance of clinical application of non-invasive PGT. So, as a modeling of a comprehensive non-invasive PGT approach should be an ideal approach. And if we look at today's application, the non-invasive PGT can be an alternative for embryo prioritization according to the haploid profile. We can give a prioritization score for each embryo. So in that way, where the embryo biopsy for the invasive method is not possible, we can give an opportunity for the ranking the embryos according to non-invasive PGT results. And of course, there are some other approaches like the embryo prediction by morphokinetic analysis. And there are also several studies has been already published. We have very good promising results. And these studies are showing very near prediction values for the aneuploid detection. But probably the most suitable approach should be the combination of these two different applications, like the genetic testing by non-invasive PGT and embryo morphokinetics, and together with some other parameters like previous clinical outcome or clinical indication of each patient. All these data if can be combined with an artificial intelligence algorithm for the embryo prioritization to select best competent embryo. As conclusion, all recent studies now demonstrating us the potential use of the cell-free DNA as an alternative to invasive PGT methods. And of course, to be able to apply in a correct way, all current IVF laboratory protocols need some modifications and to adapt for their adaptation to non-invasive PGT applications. Like each genetic test protocol, also the non-invasive PGT applications needs validation tests, and the standardization of some methods are very important, especially because today's standard invasive PGT methods are mostly designed for the amplification of targeted genomic DNA molecules from single or a few cells. But cell-free DNA molecules and their structure are totally different. And the non-invasive PGT can be used today as a scoring system for the embryo prioritization to select the embryos with higher chance of haploidy. But of course, we need some randomized clinical trial results, which are some studies are ongoing for the robust and safe, effective use of non-invasive PGT in clinical practice, and a comprehensive non-invasive screening techniques combining genetic and morphokinetic and clinical data processed by an artificial intelligence algorithm will provide us more robust data on the selection of most suitable embryo for transfer. I would like to thank my team in European Laboratory and also for collaborating centers like Dr. Veronica Bianchi, Professor Emiliano Greco, and for all other contributing laboratories to this study. And thank you for your attention.